Acinetobacter baumannii has gained notoriety in the hospital settings because the infections caused by this organism are difficult to treat. This is because these microorganisms are resistant to a number of antibiotics. On top of that, they have the ability to cause serious infections in sick patients and those with compromised immune systems. Let us briefly describe the microbiology of Acinetobacter. Acinetobacter are gram-negative or gram-variable, catalase positive, oxidase negative, non-motile, non-fermenting coccobacilli. Acinetobacter are ubiquitous in nature. They can be recovered from almost all soil and surface water samples. They have also been isolated in body lice from homeless people. So, who is at risk of acquiring Acinetobacter baumannii infection? Baumannii affects moist tissues such as mucous membranes or areas of the skin that are exposed where the integrity of the skin has been undermined. It is important to note that these skin infections can be serious. Initially, they present with a peau d'orange, then progress to clear vesicles, hemorrhagic bullae, and necrosis. If left untreated, the infection can progress to septicemia and death. Others who are at risk are of acquiring infections due to Acinetobacter baumannii. Remember, patients with artificial devices such as catheters, sutures, those who are artificially ventilated, and those who have undergone dialysis or antimicrobial therapy within the past 90 days are also at risk. So what are the virulence factors of Acinetobacter baumannii? Let's start with OMP-A, a member of the outer membrane proteins. OMP-A contribute to the virulence of Acinetobacter baumannii. OMP-A binds to the host epithelia and mitochondria and induce mitochondrial dysfunction. This is followed by the release of cytochrome C, which leads to the formation of apoptosome. These reactions all contribute cell apoptosis. OMP-A is also involved in resistance to complement and biofilm formation. The next virulence factor we will touch on is the ability of Acinetobacter baumannii to form biofilms. Acinetobacter baumannii strains are able to form biofilms on different biotic and abiotic surfaces. The bacteria in a biofilm are protected from antimicrobial and antiseptic treatments as well as host defenses. Biofilm formation contributes to the persistence of Acinetobacter baumannii in the hospital environments. The ability of A. baumannii to form biofilms allows it to grow persistently in unfavorable conditions and environments. Factors that control biofilm formation can include nutrient availability, the presence of pili and outer membrane proteins, and macromolecular secretions. Metal cations also play a role in controlling biofilm formation by enhancing the ability of A. baumannii to adhere to particular surfaces. Biofilm production represents a mechanism of resistance. It is important to note that A. baumannii becomes metabolically inert in the deeper layers of the biofilm in order to survive in unfavorable conditions. This makes it difficult for antibiotics to penetrate deeper layers of the biofilm and to act on metabolically inert bacteria. The ability to form biofilms affects antibiotic susceptibility of Acinetobacter baumannii, and this may lead to clinical failure. A. baumannii isolates might acquire multi-drug resistance within biofilm communities. This facilitates the survival and circulation chances of A. baumannii in clinical settings. The resistance of Acinetobacter baumannii to antibiotics is a public health concern. A. baumannii is one of the major agents in hospital-associated infections, especially in ICUs. The common mechanisms of resistance include the following: production of beta-lactamases, efflux pumps, decreased membrane permeability, and alteration of target site of the antibiotics. Resistance to beta-lactams is accomplished by production of beta-lactamases. A. baumannii possesses a non-inducible chromosomal AMP-C cephalosporinase. The AMP-C gene, which codes for the production of AMP-C beta-lactamases, is regulated by an upstream insertion sequence element known as ISABA1. Overexpression of AMP-C cephalosporinases and resistance to extended-spectrum cephalosporin is linked to the presence of ISABA1. Acinetobacter baumannii also has an intrinsic class D oxacylinase. This oxacylinase belongs to the OXA51-like group of enzymes. OXA51-like enzymes hydrolyze penicillins, i.e. benzylpenicillin, ampicillin, ticarcillin, and piperacillin, as well as carbapenems, imipenem and meropenem. It is important to note that the class D oxacyllinases hydrolyze carbapenems to a less extent than penicillins. Still on the beta-lactamases, Acinetobacter baumannii also possess class B or metallo-beta-lactamases, MBLs, which are encoded by mobile DNA, plasmids, integrons. These enzymes catalyze the hydrolysis of virtually all beta-lactamases, including carbapanims, except monobactams. These enzymes require either zinc or another heavy metal for the catalysis. When beta-lactamases collaborate with outer membrane proteins, it was observed that the resistance due to beta-lactamases was enhanced. In addition to production of beta-lactamases, efflux pumps also contribute to antibiotic resistance to beta-lactam antibiotics in Acinetobacter baumannii. Overexpression of 80ABC efflux pump results in carbapenem and cephalosporin resistance in Acinetobacter baumannii. Resistance to aminoglycosides in Acinetobacter baumannii is as a result of aminoglycoside modifying enzymes, target site alteration by 16S RNA methyltransferases, and poor uptake due to loss of permeability or overactivity of efflux pumps. When it comes to resistance of Acinetobacter baumannii to tetracyclines, efflux pumps are responsible for resistance. Of concern is that resistance against tetracycline has also emerged through enzymatic inactivation. These enzymes, the monooxygenases, can inactivate all tetracyclines, including the newer tetracyclines, i.e. iravicycline and omadacycline. Acinetobacter baumannii has also developed resistance to fluoroquinolones. The resistance to fluoroquinolones is via efflux pumps, alteration of target site, and can be plasmid-mediated as well. Mechanisms of colistin resistance in Acinetobacter baumannii include complete loss of lipopolysaccharide through the inactivation of the biosynthetic pathway, alteration of target site through lipopolysaccharide lipid A modification as a result of mutations in the PMR-CHAB operon and MCR genes. Thank you for listening. Please don't forget to like and subscribe to our channel